Hello, my name is Emily Ray, and this is Beth Diggle. And this summer, we worked with Dr. Mitchell in the biology department on two connected research projects, where each of us were responsible for one of them. Both of these projects were continuations of projects that we worked on last fall in Dr. Mitchell's research class. So we were both really excited to have the opportunity to continue the research this summer.
So the research project that I was focused on this summer was looking at how increasing seawater temperatures and the introduction of microplastics can impact bleaching of sea anemones, specifically Aptasia pallida. So to really understand this, I just want to give a brief background about how bleaching works. So when we first learned about this phenomenon, we looked at corals and how bleaching occurs in them. So in the corals have these symbiotic algae that live on them, and they're in a very delicate symbiotic relationship. So the corals provide the algae with protection and a stable position in the water column so that changing tides and currents doesn't affect them and have them be washing around in the water column. In addition, they also provide them with inorganic nutrients they need for photosynthesis. So in return for that, the algae provide the corals with the majority of the energy that they need through photosynthesis, which is really crucial given the lack of nutrients that are present in these marine environments. So when this relationship is disturbed by any sort of environmental factors, so that can be, like I said, increasing seawater temperature or introducing microplastics, the phenomenon of bleaching occurs. So when this happens, the corals will decrease the density of these symbionts, which is called coral bleaching. And so the algae are usually green, and when they get expelled, the corals turn white, which is why it's called bleaching. So we're hoping to see that the combination of increasing seawater temperature as well as microplastic ingestion would cause the same bleaching phenomenon in sea anemones. So we thought this was a really important issue to look at as global warming becomes more and more of an issue, and as there's more and more plastic being put into the oceans, which eventually is going to get broken down into these microplastics, which can cause issues in these marine environments. Um, so we're trying to pick up where we left off from last semester, which was just perfecting the feeding of the microplastics to the anemones. And unfortunately, that presented more of a challenge than we expected and took almost all of the summer for us to try to figure that out alone. So we did a couple of different things to try to feed these microplastics to the anemones. First, we started by putting them directly in the water and hoping that they would ingest them that way. We tried literally force feeding the microplastics themselves with a pipette into the sea anemones' mouths. And then towards the end of the summer, we decided that the best approach would be to mix the microplastics with the brine shrimp that the anemones eat and inject those into their mouth. So that one was definitely the most successful, but there's also a lot of there wasn't a lot of consistency with our results. When we counted the microplastics in the anemones, we would see some would have 40 and then others would have 1,300. So overall, this summer we kind of learned more about what not to do as opposed to getting any real results, but we also suspect there might have been an issue with the anemones we had as they weren't behaving as they usually do. For example, they're supposed to bleach at a certain temperature, and when we would put them at that certain temperature, they wouldn't bleach or they would just completely disintegrate. So again, we suspect there might have been issues there, and we're really looking forward to continuing this research with a new batch of anemones, hopefully this fall.
So while Beth did a lot of work with the anemones, a large part of my job was working with the endosymbiont algae living within the gastroderm cells of the anemones, which were Symbiodinium minutum. These algae are responsible for photosynthetically producing 95% of the nutrient matter metabolized by the anemones. And kind of the biggest part of my experiment dealt with counting the algae in culture to be able to quantify their population increase in nutrient cultures with and without sufficient phosphate for multiplication and division over like a 15-day time period. So what I did was take counts every other day for 10 days on a microscope using a counter, and then I counted every day for the following 5 days of the experiment. And while we were looking for a decrease in growth rate in the phosphate deplete population, we were looking for a constant growth rate in the phosphate replete population. That is what had enough phosphate for excess division. And while we were hoping to observe a change in cell size by looking at laser refraction for differences in light scatter, the results from both of these trial runs of this experiment turned out to be inconclusive, but they do point us in like a future direction of hoping to look at why our data seem to indicate not only not what we expected, but the opposite.